Hi, PBio. We're going to continue our lesson on linked genes. So if you haven't seen part one yet, please make sure you go and watch part one. We're continuing learning about how the idea of linked genes was developed. And we started with Thomas Hunt Morgan. Alfred Sturtevant then went on to further develop Thomas Hunt Morgan's hypothesis, which again, Thomas Hunt Morgan was developing the hypothesis that said that parental alleles are not always going to be found together because there is something breaking the pattern. And that thing that's breaking the pattern is crossing over. So he went on to say, Alfred Sturtevant went on to say, the farther apart two genes are, the more likely crossing over will occur, and therefore they will recombine more often, and they will have a higher recombination frequency. So this is the term that we're going to use and that we're going to calculate when we're talking about linked genes. So to calculate your recombination frequency, you're going to take the number of your recombinant offspring and divide it by your total number of offspring, then multiply it by 100%. This is not an equation that is on your equation sheet, AP Bio. So if you are in AP Biology and you looked for this equation on your equation sheet, it is not there. This is one of those that you're going to need to memorize and keep track of, okay? Recombination frequency is the likelihood that two alleles will end up on different chromosomes within a gamete after crossing over. So take a look at this image, all right? This is a really good image to help visualize what's going on here. We have two chromosomes. They're homologous chromosomes, meaning they have the same genes. They're going to meet in the middle during metaphase, and during that process of meeting in the middle, they're going to undergo crossing over in anaphase. That process of crossing over switches the dominant C allele and the recessive C allele. But if you notice, the A and the B alleles stayed together. So this was Sturtevant's contribution to the idea of recombination frequency, or to the idea of linked genes, is that the closer together they are, the more likely they are to remain on the same chromosome, and the less likely they are to undergo crossing over. So let's calculate the recombination frequency. If we were looking at Thomas Hunt Morgan's fruit flies, again, this is the experiment that's in your document. Which of these would be your recombinant phenotypes? Let's see what you got. If you said that the gray vestigial winged flies and the black normal winged flies were our recombinant types, then you would be correct. So these are going to be your recombinant types. So you'll divide this value by 2,300, and that will give you your recombination frequency, which if you do that, you divide it by 2,300, we've got 206 times 185, divide 206 plus 185, not times. And you divide that by 2,300, you should get 17%. So this represents a 17% chance that these traits will undergo recombination or undergo crossing over. Now, here's the thing. Crossing over is always going to happen on a chromosome. It's always going to happen. But what we are calculating when we're calculating recombination frequency is the likelihood that two genes will end up on different chromosomes because of crossing over. So that's what we're trying to determine. We're not trying to determine if crossing over occurs for the entire chromosome. We're trying to determine, does crossing over occur for these specific traits on the chromosome? So let's take a look at an example. Chromosome 1 has a recombination frequency of 10%. Now, Sturtevant also used the phrase map units or used the term map units. So if you see MU, that is equivalent to the recombination frequency. This would be 10 map units. So A and B have a recombination frequency of 10%. C and D have a recombination frequency of 47%. So, based on this information, which of these traits, which of these genes, A and B or C and D, is most likely to experience crossing over? Did you figure it out? If you said that chromosome number 2 was more likely to undergo crossing over, then you would be correct. If you look at this diagram, you can see that in chromosome 1, crossing over occurred in the same exact place as it occurred in chromosome 2. But C and D were swapped, and the dominant and recessive alleles were separated on the chromosomes. Whereas for A and B did not swap places because they moved together. They moved on the chromosome together. So they did not experience crossing over. Even though crossing over occurred, these genes specifically did not experience crossing over. Now, here's the thing. Crossing over can happen on chromosome 1. This is an example of how it could happen. It is just a much lower probability. Specifically, there is a 10% probability that these traits would undergo crossing over. So, this has to do with the idea of independent assortment. This essentially means that two unlinked genes can only have a 50% recombination frequency. Now, I want you to think about that for a second. Recombination can only be a maximum of a 50% probability. Now, why would that be? If you look at these values that I have up top, these are the values that Thomas Hunt Morgan calculated. We had a 17% recombination frequency and 83% parental types. So if you have a trait that is not linked, aka completely normal Mendelian inheritance, you would have a maximum of 50% recombination frequency. Because that means you are essentially 1 in 2 shots. You have a 50-50 shot of those traits being recombined. Okay. So if you have an 83% chance that genes will recombine, this doesn't totally make sense if you're talking about linked genes, because this indicates that two alleles will recombine more often and end up on different chromosomes, which is never going to happen. Okay. So when you're talking about linked genes, if you're doing a recombination frequency calculation and you notice that your value is above 50%, then that either means that you did something wrong, or more likely, it could also mean that you are not looking at a linked gene. You are looking at or you're not looking at an unlinked gene. You are looking at a parental genotype. So let's take a look at this. Let's take a look at some other calculations. Based on the following information, where would these genes be found on the chromosome? Now, we're going to use map units, okay? And this was the map units that you have been given. Recombination frequency can be converted into map units. So you have the cinnabar eye color, you have wing size, and you have body color. And you want to map these traits onto this chromosome. So if I were to map these traits onto this chromosome, I would want you to think about, okay, what would that look like? If I were to just put this gene, right, if this is the cinnabar gene. what would the other ones look like? So if we have cinnabar, and let's do the first one, which is cinnabar eyes and body color. And there are 9 map units between those two traits. I'm going to do right here. And then this is 9 map units between those two traits. And then you have cinnabar and VG, which is vestigial wing size. And there are 18 map units, or excuse me, not 18, there are 8 map units between the cinnabar eyes and the wing size. Then you know, because the body color and the cinnabar eyes, or excuse me, the body color and the wing size are 17 map units apart. That makes sense because if you add 18 plus 9, this would give you 17. Did I say 18 plus 9? If you add 8 plus 9, that would give you 17. And so this is how you can map units onto your chromosomes. So what do you notice about these new frequencies? There's something funky about these new frequencies. I want you to see if you can find it. Did you find it? If these new frequencies now add up to 17%, how do we go from 9.5 and 9 map units to 17 map units? Now, I will tell you this is not a mistake. This was the actual observed recombination frequencies for these traits. So why don't they add up? Well, what happened was something called a double crossover. So hang tight. I'm going to show you a diagram of a double crossover in just a moment that will hopefully help. Double crossovers result in a slight underestimate of the actual number of recombination events that would occur between two genes. So essentially what we're saying here is that recombination happened multiple times within these chromosomes. What would that look like? Let's take a look at genes A, C, and B. Okay, A, C, and B. And they're arranged on the chromosome like such. If two genes are unlinked, that means that they have a 50% chance of being on the same chromosome or ending up on different chromosomes. 50% probability. There's an equal probability of them ending up on the same chromosome or ending up on different chromosomes. That's what it means to have a 50% recombination frequency. So in this first image, recombination happened between C and B first. So what it looks like here is that now you have the dominant A, the dominant C, and the recessive B. Whereas originally you had the dominant A, the dominant C, and the dominant B on one chromosome. But crossing over happened a second time. And crossing over then happened between A and C. So now in our new crossing over or our new chromosome arrangement, we have the dominant A, the recessive C, and the dominant B. So what this looks like, it looks like A and B did not undergo crossing over, even though they did. A and B did undergo crossing over here. They went through the process. They ended up on different chromosomes. But this makes it look like that didn't happen. So this is how you can end up with funky numbers like this, where the recombination frequency for genes that are farther apart looks lower than it should be because of a double crossover event. Okay. So This is where we get to your practice problems. So this is a separate packet that you should have gotten. And because these questions are pretty challenging, I'm going to go over a step-by-step process for what you should do to try to determine the order of genes on a chromosome. Okay. First thing that you should do to determine the order of genes on a chromosome is figure out what your parental genotype is. Your parental genotype is going to be the most common genotype. Your parental genotype is always the most common genotype, which in this case is our homozygous dominant genotypes and homozygous recessive genotypes. Any other type of combination that you see is a recombinant type. So all of these other genotypes that you see are recombinant genotypes. Now, the least common genotype that you observe, that's going to be a double crossover. And that's because the double crossover means that if you go back to this example, double crossover is much less likely to happen in general. It's just statistically less likely to happen. It's far more likely that C and B would undergo crossing over, or A and C would undergo crossing over. But it's less likely they would both undergo crossing over. So this is what we're going to look at here. So, which genotypes showed you where a double crossover occurred? Well, that would be these right here, the 5 and the 8 observed offspring. So, anytime you find the double crossover individuals, the double crossover is going to show you the gene that's in the middle of your chromosome. Okay? Now, I'm going to explain why. But if you can't keep track of why, just remember, the double crossover is the least common type. And the least common type will show you which allele is in the middle. Now, in this case, we know that C is going to be the gene that's in the middle of A and B. And that's because A and B stayed on the same chromosome, but it looks like C switched. Now, if we go back to this example, this is because C underwent a double crossover event. If B was in the middle, B would have been the one that looked like it swapped places, but it's not. B is not the one that it looks like it swapped places with the others. C is, right? If you look here, we no longer have a dominant A, a dominant B, and a dominant C. We have these two that switched places. So we now know that C is going to be in the middle of our gene sequence. But we don't know how far apart they're going to be. So this is where you would calculate your recombination frequencies. So to calculate your recombination frequency between A and B, you would take the number of recombinant A and B offspring divided by the total number of offspring, which is 1,000 offspring. So anytime you see recombination between A and B, you would add that to your total number of recombinants. So this is what that would look like, okay? You see recombination between A and B 85 times. So that's this number over here. This would be 85. It occurred 85 times. The recombination between A and C. Recombination happened between these guys 81 times. And again, that's this value over here. A, B, and C. A, B, and C. So this is just a breakdown of how recombination occurred to produce all of these different types of individuals. Okay. So to determine your recombination frequency for A and B, you would add together the recombination frequencies for these 4 types. To figure out your recombination frequency for A and C, you would need to take any different combination of A and C. So this is what that would look like. I know that was kind of wordy and there was a lot of things happening there, but I just want you to look at these values. For A and C, I took 85, 81, 8, and 5, because that's where I saw recombination between the A and the C gene. For B and C, I used 27, 30, 8, and 5, because that's where I see recombination between C and B. For recombination between A and B, I took 85, 81, 27, and 30. And that's the recombination between A and B, because that's the number of times that I saw different combinations of A and B than what the parent types look like. Okay, so this is the map units that I got. I should have gotten 17.9 map units for A and C, 7 map units for B and C, and 22.3 units for A and B. And you can map these out on the chromosome, and it's in the answer key as well that's posted on Schoology. So with that, good luck practicing. I wish you the best of luck with those worked. There's some worked examples here as well if you need them, but I wish you the best of luck with your studying. And please feel free to reach out if you have any questions.